So photosynthesis in flowering plants. We are going to look at some of the neat questions, previously asked questions in the NEET in this topic. So it is very important that you should practice the questions. ATP formation in chloroplast and mitochondria is explained by which of the following theories? So we know that in case of photosynthesis in chloroplast, so this is the thylakoid membrane and a lumen, and this is the factors FO and F1, coupling factor O and coupling factor 1. So these are the protons which are accumulated here by the photolysis of water. And see, the pumping of these protons through this channel, it will lead to the formation of ATP. In case of mitochondria, the inner mitochondrial membrane. So if you imagine this as the inner mitochondrial membrane here, and there is accumulation of protons here, and then there is again a channel FO, F1. So it is the pumping of these protons here that is responsible for the formation of ATP. And this is explained by the chemiosmotic theory. So the ATP formation is easily explained by chemiosmotic theory. So that is the correct option. B is the correct option. The connecting link between light phase and dark phase of photosynthesis. So light phase and dark phase, we already know that light phase where non-cyclic photophosphorylation and cyclic photophosphorylation takes place. And this is going to give rise to NADPH2 and it will give rise to ATP. And these two are referred as assimilatory powers. These are assimilatory powers. And these assimilatory powers are used as a raw material in the dark reaction. And this dark reaction, it takes place in stroma. So the correct answer, connecting link between light and dark phase, is ATP and NADPH2. So the correct option in this case is C. The Hill reaction of photosynthesis, it takes place in out of these options. The correct answer in this case is ferricyanide. So Hill reaction is known to take place in the presence of ferricyanide. Algae employed by Calvin et al. In experiments on photosynthesis. The correct option in this case is chlorella and scandesmus. So you have to remember this. This is a memory-based question. So by using these algae, the whole Calvin cycle was elucidated. The process of formation of intermediate products in the Calvin cycle, it was elucidated by working on these algae. So the correct answer in this case is B. Which technique has helped in the investigation of Calvin cycle? So as I told you in the previous case, they have used these algae, and these algae have been radioactivated. So they have used radioisotopes. So carbon 14, etc. So radioisotopes have been used, and these radioisotopes, when they have used this radioisotopes, they have tracking the path, or they tracing the path of this radioisotope. So by tracing the path of this radioisotope molecule, they can easily find in which molecule this carbon is inserted. So therefore, the correct answer in this case is C. In C3 plants, the first stable product of photosynthesis during the dark reaction is. So because it is C3, C3 means that the compound which is formed in this case of Calvin cycle, it is a 3-carbon compound, and therefore we call this as C3 compound. So the phosphoglyceric acid, so this is phosphoglyceric acid. So phosphoglyceric acid is, out of this, it is a 3-carbon compound which is formed as a first stable product, and therefore it is referred as a C3 cycle. Correct option in this case is A. In dark reaction, the first reaction step is referred as. So in the dark reaction, we already know that ribulose biphosphate in presence of an enzyme called ribisco, it combines with carbon dioxide. And this process, we refer this as a first step, and this will give rise to phosphoglyceric acid, two molecules of phosphoglyceric acid, which is a 3-carbon compound. So this reaction where ribulose biphosphate combining with carbon dioxide, we call this as carboxylation. Dark reaction of photosynthesis is also called so because now the reason why the dark reaction is so called is because it does not require light. Therefore, we also call this as light. We also call this reaction as light independent reaction. We also call this as light independent reaction. That means the dark reaction which takes place in the stroma of chloroplast, and it does not require light. The dark reaction, it depends on the products of light reaction, but by itself, it does not require light. So it is also referred as light independent reaction, and therefore the correct option in this case is does not require light energy. The initial enzyme of Calvin cycle. The initial enzyme of Calvin cycle, we call it as rubisco. Rubisco is the correct option. What is this rubisco? It is ribulose. It is ribulose biphosphate carboxylase. And this combines with ribulose biphosphate. Ribulose biphosphate combining with carbon dioxide, and therefore we call this as the enzyme is represented as rubisco. So rubisco option A is correct one. We go to the next question. In C4 plants, C3 cycle occurs in which of the following? So in C4 plants, one, there is a mesophyll cell and there is a bundle sheet cell. So this is referred as a mesophyll cell and this is a bundle sheet cell. So it is in this bundle sheet cell that C3 cycle takes place. So in the bundle sheet cell, it is the stroma of bundle sheet cell that C3 cycle takes place, because in this case, in the stroma, there is presence of an enzyme rubisco in the bundle sheet. Correct option in this case is A. A molecule of glucose is formed in Calvin cycle from. So for the formation of glucose, the options given are these out of these following options. Which is the correct option? So is it 6CO2 12 ATP, or is it 6CO2 18 ATP 12 NADPH2? So we know that these are the assimilatory powers. These are the assimilatory powers, and these originate or these come from the light reactions, and this is the carbon dioxide which is fixed. So if you look at the proportion, it is 6 molecules of carbon dioxide with 18 ATP molecules and 12 NADPH2 that will result in the Calvin cycle with the formation of glucose molecule. So the correct option in this case is B. For assimilation of one CO2 molecule, the energy required in the form of ATP and NADPH2. So this Calvin cycle, it runs 6 times. We already know that it runs for 6 times. So if we divide this number, in the previous question they have given, if we divide this number, so 18 divided by 6, so that much number of that amount of 3 carbon dioxide as 3 ATPs, and if you divide 12 by 6, you will get 2 NADPH2. So this is per this value is for 1 carbon dioxide. So per carbon dioxide molecule. So the correct option in this case is for assimilation of 1 CO2 molecule. The energy required in the form of ATP is, it is 3 ATPs and 2 NADPH2. Correct option in this case is A. The correct option in this case is C. 3 and 2. So this is not the correct. C is the correct option. For the synthesis of 1 glucose molecule, the Calvin cycle operates for, so it runs for 6 times, as I have told you previously. So 6 times, that is 6 molecules of CO2 are fixed. So we say that it runs for 6 times. So correct option in this case is C. Phosphoenol pyruvic acid is a primary carbon dioxide acceptor. So this PEP pyruvic, this is a bundle sheet, this is a mesophyll cell, this is a bundle sheet, and this is C4 plants. So phosphoenol pyruvic acid, it accepts carbon dioxide here in the mesophyll cell. And the enzyme required is PEP carboxylase. So therefore, in which type of plants this PEP accepts carbon dioxide? So this is the characteristic feature of C4 plants to minimize or to reduce the possibility of, so this will reduce the possibility of photorespiration. Very important point which one has to remember in this case. Bundle sheet cells are, so here the bundle sheet cells, these bundle sheet cells, they do not have PEP carboxylase. Instead, they have rubisco. So these are rich in rubisco, and therefore C3 cycle takes place in this. So the correct answer in this case is D. Assume a thylakoid which is somehow punctured so that the interior of thylakoid is no longer separated from stroma. This damage will have most direct effect on which of the following process. So this thylakoid lumen which is present here, this thylakoid lumen is crucial. This is important because it maintains something called as a proton gradient. And this proton gradient is important for the formation of ATP. So when these two protons are pumped outside at a time, then there is a formation of ATP molecule. So if it is punctured, these protons will not be pumped. And if there is no pumping of the protons, ATP will not be formed. So this will directly affect the synthesis of ATP. So in this case, the correct option is in cyclic photophosphorylation, the electron released by the reaction center P700 is accepted by. So if it is P700, so it will be an electron acceptor. Here there is ferredoxine. Again, it reverses back. So eventually, finally, ultimately, they have asked in the question. So read it very, very carefully. So in cyclic photophosphorylation, the electron released by the reaction center. So when the electron is released from here, it travels back in a cyclic process, and therefore the correct option in this case is the reaction center itself. So electron is released, ejected here, and it comes back here. So therefore, correct option in this case is C. In the given diagram, you have to identify what is A, B, and C. So with A, B, and C, so this is cyclic photophosphorylation. So in a cyclic photophosphorylation, the photosystem, it is P700. So this has to be P, this has to be PS1. And this chlorophyll is P700. And this here is an electron acceptor. So we have to match this. So A is PS1. A is PS1, B is electron acceptor, and C is P700. So correct option in this case is B. chemiosmosis requires. So if you look at this chemiosmosis, again proton pumping takes place through this F0, F1 factors, or we call it as coupling factor 0 and coupling factor 1. So by the pumping of protons, what is formed? ATP is formed. What is required? Membrane is required so that gradient proton concentration can remain. So this is one of the important components required. Proton pumping has to take place, otherwise ATP will not be formed. And because of accumulation of proton, that is proton gradient is also required for ATP. So all these three options are required for the formation of ATP, and therefore the correct option in this case is D. In light reaction of photosynthesis, the chlorophyll is subjected to. So when the chlorophyll molecule, say for example P680, so when the light falls on this, the electron, it is excited and the electron is given out. So that process is called oxidation. And this chlorophyll molecule is said to be oxidized. Now this is responsible for splitting of water molecule, and because of that protons are generated and electrons are generated. So this chlorophyll molecule, it will pull electrons from this water, and therefore it undergoes a reduction. So first it will lose electron in a process called oxidation, then it will gain electrons in the process called reduction. So in this case, the correct option is both oxidation and reduction. C is the correct option. Which of the following is correct about photorespiration? So photorespiration, it takes place in presence of large amount of concentration of oxygen. So in this case, rubisco, it acts as oxygenase. So oxygenase, which is responsible for the diversion of cycle from C3 to C2. C2 cycle is also referred as photorespiration. So because of high concentration of oxygen, rubisco now acts as ribulose biphosphate oxygenase. And therefore, what are the characteristics of these photorespiration? It is a wasteful process, yes, because it does not lead to the formation of glucose. So glucose is not formed. So this is a waste process. It is energy consuming process. It requires ATP, and it also releases carbon dioxide. So carbon dioxide is also released in this process in the mitochondria. So therefore all these statements are correct. So the correct option in this case is B. The photosynthesis of in photosynthesis, oxidation of dash and reduction of dash takes place. So oxidation in this case, where this hydrogen and oxygen, they are separated, so oxygen is released, and reduction where this carbon dioxide combines to form glucose. So the correct option in this case is water and carbon dioxide. Crans anatomy is a characteristic feature of the leaves of. So this crans anatomy is referring to C4 plants, and these C4 plants, they are found in the tropical regions where this has high temperatures. And the best example in this case is sugarcane. So out of the given options, the correct option is option C. The process which makes a major difference between C3 and C4. So between C3 and C4, C3, in this case, if there is an excess of oxygen, if the concentration of oxygen increases in C3 plants, then this will be diverted to photorespiration. But in this case, there is a mechanism, we call it as carbon dioxide concentrating mechanism. So because of this carbon dioxide concentrating mechanism, there is no photorespiration which takes place in C4 cycle, whereas in case of C3, there is a possibility of photorespiration. So the correct option in this case is photorespiration. Which of the following cells of C4 are prominently loaded with starch. So again, if we go to the diagram of C4, we have a mesophyll cell here, we have a bundle sheet cell here. So first carbon dioxide fixation takes place here, malic acid is formed, malic acid enters inside, it will release the carbon dioxide. Here a Calvin cycle takes place and the final product of glucose will be formed here. And this glucose will undergo polymerization and it will give rise to a starch. So a starch will be stored here in the bundle sheet. So the correct option in this case is C. So these were the set of 25 questions we did here and which were asked in the previous NEET examinations. So you have to continuously solve these questions, revise these questions so that you will get the accuracy and speed. We will continue with few questions in photosynthesis in the next session. Thank you.